Hi everyone, my name is Elizabeth Brower. I'm one of your instructors for Plant Immunity. And today I just want to introduce plant pathology history and some disease cycles as part of your module 1.2. So really quickly, symptoms can involve many different types of visual phenomenon. In general, when we look at plant disease, we're thinking about reduction of yield or biomass production by plants. But we can also see things like chlorosis, which are these yellowing spots, necrosis, necrotic spots, which are inductive of cell death, wilting, which is when a vascular system is cut off, so water is no longer reaching the leaves of the plant, or mottling, which often occurs with viruses. We can see galls or canker formation, various different types of fuzzy necrotic lesions, phyllody, which is basically when the pathogen takes over a development of part of a plant and converts flowers into leaves, for example, and of course, discoloration. So I want to get a little bit into plant pathology history because I think it gives you a good idea of the types of tools that we've developed as a field. There's 4 basic areas of plant pathology history, and these are outlined in the review as shown below. So check it out if you're interested in looking at more. But in general, there's the pre-1900s were characterized by noticing connections. 1901 to 1984 approximately is when the disease physiology era reigned supreme. Then we had molecular biology, which emerged in the early 80s to the 2000s, and then genomics and systems biology, which is the current era that we're in now. So noticing connections, I just want to mention that most of the history from this period is from Europe, because there's a lot of written records from that time, but certainly there's a very long history of humans noticing that plants are being infected. And so we can't cover all of that. But in general, we've noticed that in the 1600s, we started to see that French farmers took note of something called wheat stem rust. And they noticed that that occurred close to barberry bushes, which are these basically weedy bushes that grow on the periphery of wheat fields. And so as a group, they actually convinced the government to get rid of barberry bushes, and that really increased or decreased the amount of wheat stem rust that they saw. We also started to see that ergotism, for example, one of the diseases that was occurring in the Middle Ages, seemed to follow when after you'd consumed ergot-infested grains. And so what that looks like is here. These are the sclerotia of an ergot. So they're a fungal pathogen that are producing these fruiting bodies in the grains. And when you grind this up and make it into flour and consume it, you experience hallucinations, some of which are thought to contribute to the Salem witch trials. Around this time, we also had some big steps in microbiology and biology in general. So we saw invention of the microscope. We saw development of nomenclature and phylogenies, so essentially trying to classify organisms in relation to one another by Charles Darwin. And that paved the way for basic microbiology in the 1700s. So Pier Antonio Micheli in 1729 described all kinds of different fungal structures because he had a microscope and was trying to just map out what was out there. But what he actually did was he took pieces of the fungi, so their spores or their hyphae, and he put them onto healthy melons, and he was able to infect melons and cause disease. So he proposed the idea that you can cause disease from these very small microscopic pieces of a fungus. And that was not accepted at the time because the idea of spontaneous generation prevailed. So that's the idea that fungi or disease in general seems to spontaneously arise from nothing, from air, basically. It was only in 1861 to 1863 that Anton de Berry proved that potato late blight was actually caused by fungus. And around the same time, Louis Pasteur proved that microbes were produced by microbes and that infectious disease is caused by germs. So one of the reasons that this arose was because the idea of disease is very prevalent in people's minds around this time, both because of the really terrible medieval diseases that occurred in humans, but also because of things like late blight, potato late blight, which caused the Irish potato famine. So this occurred in 1845, 1846, and a few years after that as well. And what was happening at the time was in Ireland, they were completely dependent on producing potatoes as their staple crop. So potatoes were easy to grow in this environment. The farmers could actually take potatoes from one season and plant them in the next season, so they didn't have to buy a lot of inputs in order to grow potatoes. And they're quite poor because they rented land from absentee English landlords. And so they could also get a lot of nutrients that they needed from potato, like vitamin C. So as a result, they were very dependent on it. They grew monocultures, and it just so happened that a pathogen, an omycete called Phytophthora infestans, happened to be in the area at a time when the environment was conducive for that particular pathogen to take hold. And this pathogen is absolutely devastating. It can destroy fields within 2 to 4 weeks. And so it was a very dramatic decline, and one that was very much unforeseen and not understood. Unfortunately, at that time, because of the political climate, the British refused to help the starving Irish, and that led to starvation, secondary diseases because of malnutrition, and then mass immigration to North America and other parts of the world. So at this time, Miles Berkeley had been observing this disease. He had identified some of the spores from that infection, for example, and he was able to show that he could induce the disease in healthy plants just by applying the spores. So again, he proposed the idea that microbes are actually causing the disease, but it was not believed, unfortunately. So after it was generally accepted that microbes do cause disease and that we can monitor them under microscopes, the era of disease physiology really exploded. So this consisted of basically describing diseases, culturing pathogens, and developing the protocols to do that, looking at symptoms and describing them, and understanding the variation that occurs in plant resistance. There was also a number of chemical treatments that were produced at this time. Copper sulfate, for example, is still used today in organic agriculture. Systemic fungicides and antibiotics were discovered around this time as well. And you can see it makes a huge difference, some of these mixtures, and still do today. It was also a time of really strong biochemistry as a tool. And so what people would do is often grind up necrotic lesions like you see here, or sunken lesions. They would fractionate the molecules within that slurry, and they would try to find the specific enzymes or molecules that would produce the visual symptoms that they were seeing on diseased plants. And so this allowed people to discover a lot of cell wall degrading enzymes that are basically used by necrotrophic pathogens or hemibiotrophic pathogens to kill tissue very effectively. So they basically degrade the cell wall of the plant cell, and they burst open the cell. And that allows the pathogen to feed on the contents of the cells. We also saw host-specific toxins, and we also developed things called disease cycles. And this is just a visual representation of the entire life cycle of an organism or of a plant pathogen. So this specific example is from the wheat stem rust that I was describing before. We're not going to go through this in detail right now, but essentially there's different spores that it produces, and it produces one type of spore on wheat and a different type of spore on barberry. So to complete its entire life cycle, and be able to develop new genetic modifications or changes, it has to go through between these two hosts. And so by eliminating this barberry plant, we've actually been pretty effective in limiting the amount of wheat stem rust that we can see in some regions. Also at this time, we developed etiology. So this is basically trying to assign a specific microbe as a causal agent for a specific disease. And this, of course, holds true not only in plants, but also in animals. And so what you try to do is observe differences between healthy and diseased organisms. You look for a potential microbe that could be causing the disease, and then you have to extract it and purify it, isolate it and culture it, so that you have the one causal agent as opposed to lots of other contaminating organisms that could be on the surface of your host. So once you have this pure culture, you inject a healthy host with it, and you look for the disease. And then you can extract the organism again. Another important concept that came about at this time was the gene-for-gene hypothesis. So the gene-for-gene hypothesis is really referring to the host resistance. And what we do in this case is basically take a resistant parent, a susceptible parent, and we cross them together, and then we grow up the progeny of this cross. So all of the children from those two parents, and we observe whether they show resistance or susceptibility. So in this population, half of the plants are completely susceptible and half are resistant. We also see cases where we have a whole gradient of susceptibility to resistance. And we call these two scenarios qualitative resistance and quantitative resistance. And Harold Floor in the 1940s, when he was studying this flax rust disease, was able to hypothesize that there are single genes involved in qualitative resistance. Based on our knowledge of Mendelian genetics, the way this segregates, it implies that one gene is responsible. And we call the resistance gene that segregates in plant populations a resistance gene, or an R gene. This provides the qualitative resistance. An avirulence gene is the pathogen equivalent, or is basically recognized by the R gene. So you need to have the pathogen avirulence gene there to actually have this type of resistance. Quantitative resistance actually is multigenic in nature, just because of the way, again, genetic segregation works. And again, this is showing basically the solution to the problem, that indeed this is true. We have single genes responsible for resistance and multiple genes responsible for resistance. But we didn't understand why. We didn't understand the mechanisms of how that actually worked until the molecular biology era. And this is in 1984 approximately to 2000s. So the molecular biology era is really characterized by the fact that we've developed tools at this point to understand what genes are, what they do, and we can manipulate them. So that's key, because a lot of the approaches during this era are to produce mutations in genes or knock out genes, which basically means reducing expression or overexpressing genes. And then we're screening libraries of plants that have mutations in them in an unknown gene, and basically looking for, again, susceptibility or resistance. This can be applied to either the pathogen side or the plant side. And in the pathogen side, there was a big movement at this time to identify something called a virulence factor. So pathogen virulence factors are the genes that enhance the pathogen's ability to cause disease. So when you get rid of these genes, you see a reduction in the amount of disease that you will get, but the pathogen can still infect to some extent. We also saw pathogenicity factors start to arise. And so these are genes that you need to have in order, as a pathogen, in order to infect at all. So when you don't have these genes, you cannot infect. This is sort of an all or nothing type of response, whereas this is a quantifiable difference in the amount of disease. So interestingly, the genes we discovered in this era as being virulence factors or pathogenicity factors were not the same genes that we identified in the previous era, the cell wall degrading enzymes, host-specific toxins, likely because there are so many functionally redundant versions of these types of compounds that knocking one down or getting rid of one gene doesn't actually show you a big difference. So make a long story short, and again, we'll go into the details of this. We discovered that there are two different layers of inducible immunity, and that these underlie the qualitative resistance and quantitative resistance that we've seen in the previous era. So ETI, or effector-triggered immunity, is basically triggered by recognition of R proteins, or R genes, of those avirulence factors. And avirulence factors can be effectors, which are proteins just secreted by a pathogen into the plant cell. Effector proteins, by definition, may or may not be virulence factors or pathogenicity factors, but in general, they're thought to target the host responses or cell function or some aspect of plant disease or defense or immunity. So quantitative resistance is characterized by a number of different things. And again, we'll get into different forms of resistance. But in general, it also includes recognition by the plant of microbe-associated molecular patterns. So these induce pattern-triggered immunity, which we'll call PTI. But these are basically small molecules that are given off during infection inadvertently by the pathogen, and those can be recognized on the periphery of the cell. And again, we'll get into the mechanisms of that, but the important thing to know is that gene-for-gene interactions are mediated by ETI, and quantitative resistance is encompassing all kinds of different forms of immunity. So throughout this time period, we also developed basically Cox postulates in reference to specific genes. And so Stanley Fockow proposed this in 1988. Basically, the genes under investigation has to be associated with pathogenic members of a genus or pathogenic strains of a species. And this is a reference to trying to look at pathogenicity factors or virulence factors. So if you think a gene should be important, then it should be present in pathogenic members of the strains of a species. It should also be produced during infection by the pathogen in a location in the host that's appropriate for its proposed function. When you knock out the gene, you should be able to measure some change in pathogenicity or virulence, and reversion or replacement of the gene should restore that pathogenicity or virulence. Okay, so we were basically able to understand a lot of the mechanisms underlying disease resistance through those types of approaches. But currently, we're in the genomics and systems biology era, and we still use those previous approaches, but we've added in a couple of new approaches because of the availability of genomic sequencing and the availability of bioinformatic tools that allow us to combine large data sets. So for example, we can systematically compare plant or microbe genomes. So here's 4 fusarium genomes that were completely sequenced and compared. And what they do is they map out where genetic mutations lie, and they overlay that on basically the proteins that they know are secreted. So which regions of the genomes correspond with that, which ones are actually produced in the plant during infection, which ones are translated. And this gives you an overview of, like the result of this study was basically to show that the genes that are involved in virulence that we know about actually occur in these these regions containing a lot more mutations. So it seems that certain areas of pathogen genomes are mutating at higher, faster frequencies than others. And that tends to associate more with virulence as a way of potentially producing new types of virulence factors. So here's an example of systems biology. Systems biology is basically a way of looking at the emergent properties of a system by analyzing many different components within that system. So for example, instead of trying to look at one specific gene at a time, you may be looking at an entire gene family. In this case, we have protein kinases. So we knocked out all of the members of this family that we could, and knocked out several different effector proteins in the pathogen. And a combination of the physiological traits that we could find as a result of these interactions allowed us to create a network, basically co-occurrence for specific genes and their functions in plant immunity. So this is a very sort of math-heavy way of trying to look at, get a broader perspective of the entire immune response as opposed to a specific gene within the immune response, because the idea here is that ultimately organisms function as a unit of many different genes and proteins and processes. And so looking at one gene at a time is not necessarily going to give you the whole picture on what's going on during infection. So I'll leave it there for now, but we're going to have a discussion on Friday, and we'll get a little bit more into what we're expecting for the class. So I'm really happy to go through any of these concepts again, if any of them are unclear for you. So with that, I will talk to you later.